Welcome back to Divan's Biology. My name is Dr. Aruna Divan and today we are going to talk about homologous and non-homologous chromosomes. First of all, we will understand the basic concept of homologous and non-homologous chromosomes, and then we will summarize the topic by distinguishing between both of them. You know that the word chromosome is actually based on a word chroma. What does this chroma means? Chroma is a color, right? Now why color? Because chromosomes are the long thread-like structures that has a spatial affinity towards basic dyes. Okay. Now what about homologous chromosomes and non-homologous chromosomes? To understand homologous chromosomes, we need to start from the scratch. Okay. Now we know that a diploid cell has two sets of chromosomes, right? Let us suppose this is one set of chromosome and this is another set of chromosome. Okay, let us make another set of chromosome by different color. This is another set of chromosome. Okay, how many chromosomes are these? These are 23 chromosomes. These are 23 chromosomes which are contributed by father, or known as paternal chromosome, and these are the 23 chromosomes contributed by mother, known as maternal chromosomes, right? Now these 23 chromosomes, the set of this 23 chromosome is known as a genome. So it becomes very easy for us to understand what is a genome. It is just one set of 23 chromosomes. Whether it is paternal chromosome or maternal chromosome doesn't matter, but if there is a set of 23 chromosome, it is a genome. Okay, now in a diploid cell we have two sets of genome, hence we say it as a diploid cell or 2n. One set of genome is considered as X or denoted by X or small n. Here we have two sets of genome, so we can say it as a diploid cell. Similarly, if a cell have three sets of chromosome, then it will be said to be a triploid cell. Okay. Now we know that in the case of meiosis, a parent cell gives rise to daughter cells. But in case of meiosis, the daughter cell would be having half the number of chromosomes. In the last video, we talked about meiosis. Now, in that video, we came to know that the gametes thus formed by meiosis contains one full set of chromosome, that is 23 number of chromosomes. Not 46. DNA is having 46 number of, a diploid cell is having 46 number of chromosomes, but this is a haploid cell. Either this is a gamete, male gamete. Let us suppose this is a male gamete. Okay, this male gamete is having 23 chromosomes. Now these 23 chromosomes, these two chromosomes or this chromosome with any other of the chromosome, they doesn't match at all. They are not similar. None of the chromosome is similar to the other one in this 23 set. Now suppose this is another set of female gamete. Female gamete means either egg or ovum, right? Again this is having a set of 23 chromosomes, not 23 sets of chromosomes, a set of 23 chromosomes. Then these chromosomes doesn't match with each other at all. Okay, when male gamete and female gamete fuse, what do they form? They form a zygote. Now again this zygote is having two sets of chromosomes, right? One set contributed by male gamete and one other set contributed by female gamete. Now zygote is again diploid and it is having 46 chromosomes. Of these 46 chromosomes, 23 are from mother and 23 are from father. Now let us come to the homologous pair of chromosome. What is this homologous pair of chromosome? Let us suppose this is a zygote, right? This zygote is having how many chromosomes? This zygote is having 23 chromosomes from mother side and 23 chromosomes. Let us suppose this is 23 chromosomes from father side. Now what will happen during meiosis 1 in prophase 1 in meiosis 1? The chromosome number 1 of mother will start looking for the chromosome number 1 of father. Why? To form a pair. Okay. Chromosome number 1 of mother will form a pair with chromosome number 1 of father in a zipper fashion. All these things are explained in detail in meiosis. Number 1 of mother has formed a pair with number 1 of father. Number 2 chromosome of father is here with number 2 chromosome of mother, and likewise 23 chromosome pairs are formed. Okay, now this pair is known as a homologous pair. This pair is known as homologous pair. But this and this, if they form a pair, then they will not be homologous to each other. They will be non-homologous chromosomes or non-homologous pair of chromosomes, right? Of these 23 pairs of chromosomes, we know that 22 are autosomes, and the 23rd pair, it can be X or X in case of female, and it can be X and Y in case of male. So these 22 autosomes and this one pair, X and X, these are homologous pairs, but this is not a homologous pair. Now let us understand why we are saying that this is a homologous pair, this is a homologous pair, but this is not a homologous pair. There might be some reason behind it. Okay, let us understand that reason. Let us suppose this is a chromosome contributed by father and this is a chromosome contributed by mother. Okay, this is the centromere position. This is the centromere position. Now what are the special characteristics of homologous pair? The first one is that homologous pair have more or less similar length. Let us suppose this is first pair of chromosome and let us suppose this is second pair of chromosome. Chromosome number 1 of father, chromosome number 1 of mother, chromosome number 2 of father, chromosome number 2 of mother. Okay. Now the main characteristics that tells us that this is a homologous chromosome, the first one is the length of homologous pair is more or less same. Secondly, the position of centromere of homologous pair is similar. We know that the chromosomes are divided into 4 types on the basis of this position of centromere that I have already discussed in meiosis, my previous video meiosis. Okay. Now the second characteristic is the position of centromere of homologous chromosomes is always same. Let us make it again to make it more clear. Okay, this is second pair. Now you can see the centromere position of this is same, but the centromere position of this is not same to this. If we try to form a pair of these two, these would be non-homologous chromosomes, because first of all the length is different and secondly the position of centromere is also different. How can they make a pair? Okay, if they are going to make a pair, then we can say this is a non-homologous pair. The third characteristic of homologous pair is the position of gene. The sequence of gene is always same in homologous pair. right? The locus of gene, locus means position of gene, it will also remain same. Let us suppose this is the locus or position of gene of father's eye color, father's eye color, okay. Then in mother's homologous pair, this would be the locus, the same locus would be there for mother's eye color, mother's eye color, okay. Now here the genes are similar but not identical. Why? Because let us suppose father is having black eyes and mother is having blue eyes, then what will happen? Then we can say that these two are similar but they are not identical, okay. So the third one is that gene sequence is same, the locus of gene is same, while this is different in case of non-homologous chromosomes, okay. These homologous chromosomes, these homologous, in the video of meiosis you might have seen that these homologous chromosomes when gets replicated in interphase, after that in prophase 1, in case of meiosis these start crossing over, these two start crossing over with each other, right? When they start crossing over with each other, first they form pair. In case of meiosis, these homologous chromosomes form pairs, and then these homologous chromosomes starts crossing over with each other and genetic variation is done, okay. Genetic variation is found because of the presence of this crossing over in meiosis. But sometimes what happens, sometimes due to non-disjunction, when non-disjunction of this pair happens, then what will happen? Due to the non-disjunction, sometimes one gamete will receive only one chromosome and the other gamete will receive 3 chromosomes, right? This is the chromosomal abnormality. This is known as monosomy and this is known as trisomy, okay. Now the most common example of monosomy is monosomy X, means a baby is getting only one X chromosome while the other gamete might be having whether it can be Y or it can be X, but these are all obviously X. This is monosomy X. The second common one is trisomy, that is trisomy 21. This monosomy X is also known as Turner's disease or Turner's syndrome. This trisomy 21 is known as Down's syndrome, right? It means in number 21 chromosome, we know that we have 23 pairs of chromosome. In 21st pair there is non-disjunction and the baby is getting 3 chromosomes instead of 2. So this trisomy 21 is also known as Down syndrome, in which gradually the child will develop developmental and intellectual problems. Now let us summarize the topic by differentiating between the two. Okay, this is homologous chromosome pair and here we will write the characteristics of non-homologous chromosome pair. The first difference between homologous and non-homologous chromosome pair is that homologous chromosome pair, the length of the homologous chromosome pair is approximately same. Let us suppose this is chromosome contributed by father, this is chromosome contributed by mother, so this will be, the length of both will be almost similar. Okay. Second one is same centromere position. Here we can see the centromere position of this chromosome pair. Okay, the centromere position is more or less same. According to the centromere position, according to the position of centromere, the chromosomes are divided into 4 types: metacentric chromosome, submetacentric chromosome, acrocentric chromosome, and telocentric chromosome. Here we can see the metacentric chromosome. Okay, a metacentric chromosome means the position of centromere is at the middle of the length of the total length of the chromosome. Here in this case, the length of homologous pair is more or less similar, but if we see this, then the length of this and this is not similar, so these are non-homologous chromosomes. The next one is gene sequence. The gene sequence, I have already discussed, the gene sequence of homologous pair is same and the gene locus is also same, while in case of gene sequence and gene locus of non-homologous chromosome is different. So the approximate length of the homologous chromosome is same, but approximate of length of non-homologous chromosome is not same. Centromere position of homologous chromosome is similar, but centromere position of non-homologous chromosome is not similar. The gene sequence of homologous chromosomes is same, but gene sequence of non-homologous chromosome is not same. The locus, similarly in locus, the locus of gene in non-homologous chromosome is not similar. Now the next one is homologous chromosome pairs during meiosis, but non-homologous chromosome never pairs during meiosis. Okay, the parts of the homologous chromosomes are exchanged during recombination. The parts of homologous chromosomes are exchanged during recombination. Now what is this recombination? This recombination is the time when the genetic material is being exchanged between the two homologous chromosomes. But when two non-homologous chromosomes exchange their material, then it is known as translocation. Now this translocation and recombination, both these give a genetic variability, right? The simple example, the common example of homologous chromosome pair is 22 autosomes as well as XX chromosome pair, while non-homologous chromosome pair example is XY chromosome. I hope everything is very clear to you. Thank you.